So you've probably seen the movie Jurassic Park or one of the many sequels. If you think back to one of the first movies, they talked about this idea of how they made the dinosaurs, right? So in theory, there were these ancient insects that were biting and feeding on ancient dinosaurs, and in that what they ate in their blood, right, we'd find some dinosaur DNA, and then they took that, did some cloning and put it in a different egg, and all of a sudden then we got dinosaurs, right? So the science was a little sketchy on that. Is something like this maybe possible in the future? Perhaps we'd have to have a complete set of DNA, but it brings up some interesting issues of cloning. Should we be cloning ancient animals that are no longer alive, that have gone extinct? So that's what we're going to talk about in this video. We're going to talk about the process of cloning, and we'll get a little bit into the process of cloning humans and maybe the process of cloning extinct animals to bring them back. Let's get started. So let's start with some definitions. What is cloning? Basically, cloning is producing an organism that has identical DNA, right? So if you think of humans, we don't really go through a cloning process. We go through a sexual reproduction process where you are half your mom and half your dad, right? A clone would be 100% an exact copy of another organism. So if you think about it, in humans, obviously, we aren't cloning yet, but we clone many other organisms. In fact, many of the way that we propagate plants, that is, we take certain trees and make more trees. Apple trees are a prime example, right? We don't take an apple seed, put it in the ground. What we actually do is we do a cutting from a particular plant, and then we can graft it onto a different stock. Here, what you see is some grapevines where we're taking some cuttings and then propagating those plants, where the resulting grapevines are actually all clones of the original grapevines, right? So that's how some of these vineyards keep their particular taste or particular grapes throughout many generations, because they're just clones. Okay, so what's the process of cloning? Well, I'm going to use the process of cloning animals, right? I just talked to you a little bit about how you might do that in plants. Animals, it's a little more tricky. So if we wanted to clone, for example, this sheep here, what we could do is we could take, for example, any cell, but in this case, we're taking a mammary cell. And what we do is we remove the enucleated cell and we take out the nucleus. Well, do you remember what's in the nucleus? Of course, the DNA, right? So we take out the nucleus. Well, what are we going to do with that? Well, then we could take an egg cell donor. So this is from another sheep, for example. We take one of their egg cells that has not been fertilized, right? So there has been no male to fertilize this, no sperm. We're just taking the unfertilized egg cell, taking out the nucleus of this, but in this case, we're going to get rid of the nucleus. Well, this over here is a fully fledged nucleus with 100% of the DNA. So if we're taking this nucleus and then put it into that enucleated egg cell, you can see that we now have a fused cell, which is an egg cell that was taking the nucleus out, but then we put in a fully formed nucleus from another cell, one of our normal cells, and then we let that grow, implant into an embryo, right? We can do this in a tissue culture, and then we implant it into a foster, and then that foster would give birth like they were giving birth to their own child, but it's actually a cloned lamb from this nucleus donor, okay? So in humans, it would be a little more complicated, but the basic principle is there, where we can take the nucleus out of something, right? So this animal, this particular sheep, as you can see, is fully formed. So if we take some of its cells, that's what we got going here. So we wouldn't need its sperm or egg, right? Because we want one of its fully, what we call diploid, 2 sets of chromosomes, so that we have the full nucleus intact. Okay? So now I want to go a little bit over what have we cloned before? All right, so it started pretty easy with, well, not easy, but it started more simplistic with just cloning molecules. And we figured out how to clone molecules and how to clone DNA, and we have actually a lot of biotech tools that help us do that. Then we started moving towards animals, to different types of organisms that are a little more complex, like the tadpole we did in 52, mice in 86. Then, big thing, which I'll talk about more, in 1996 is when we started moving towards bigger mammals, and I'm going to show you the process of cloning Dolly here in a moment, the sheep. 2000 we did a rhesus monkey, then cattle, cats, dogs, rats, horse, and then primates. All these pictures you are seeing here are actually cloned animals, right? So here's the clone from there. Here's some cloned monkeys. Here's the cloned camel, right, from its clone donor. And so we've actually already cloned a lot. So you may say, well, what's the difference? Isn't this the same as a baby? Well, remember, let's use this dog. If this dog was just a baby from the mother, remember it would be half this mother and half of the father. But because we've cloned, we've actually just taken the entire DNA from the mother and gone straight to the child. Okay, so that's like getting an exact replica of an organism. Okay, so I said I'd mention Dolly here. Dolly was our first cloned mammal, and I remember when they did this in 2003. It was a big deal. You can see it was on the cover of all the magazines. We read magazines back then. And there's a picture of Dolly. What we did was we took an adult mammary cell from a sheep, and we took that cell, and like I showed you before, we took two different runs. So here is our nucleus donor. This is the one that's going to get cloned. So if you look at Dolly down here, Dolly's a clone of this. So we took that mammary cell, took out the nucleus, right? And then we took another sheep, or a ewe here, right? The Scottish blackface, which is actually donated the egg and the cytoplasm, right? Took out the nucleus, and then using some pulse, we took the nucleus from this one, implanted it into the cell from here, and then this can grow, and then it starts going through all of these development processes. We called this one right here a blastocyst. When it gets to having several of these cells from the culture, we can actually then take that and put it in a surrogate, and this surrogate will then birth Dolly. Notice this surrogate was not the same type of sheep, but Dolly was this sheep from the donor there, the Finn Dorset. Okay? Just interesting. Why do we call her Dolly? Because we got this from a mammary gland, right? Like from the breast's mammary. And of course, Dolly Parton was and still is a national treasure, but she was pretty popular back then. All right, so this really started the debate about the ethical cloning of mammals. Should we be doing this? Should we be making these clones? All right, so then another sort of ethical thing comes up. What about cloning endangered species? Not necessarily even species that have gone extinct, but what about the ones that we as humans have caused to become endangered? One of the species that's actually become extinct was the woolly mammoth. In fact, there has been a joint Russian-Japanese team, and they're still kind of working on it as far as I know, that's been trying to extract enough DNA from frozen specimens that we have found. So the idea is to get a full set of DNA from this woolly mammoth implanted into some sort of other cell, maybe an elephant, and then see if that can be born, and we could bring back a woolly mammoth. Up to this point, the attempts have failed at this, but we will eventually probably get there where we are able to bring back not only extinct animals, but maybe save some of the endangered ones. In fact, we've actually now started, there's a few of these frozen zoos where we take tissue from the world's most endangered rare animals, right? So in case they do go extinct, well, we've got the tissue, we've got full sets of DNA that we could actually clone and bring some of them back if we decided we wanted to do that. Okay? So let's now talk about a little trickier topic, human cloning. And when we say human cloning, you've got to think it's got to go through the same process. We're not taking an adult human, sticking them in a copy machine and making another adult human. If we were to clone, we'd take the cells like we did with Dolly, implant them in another egg, and then implant it in a surrogate mother, then that would be a little baby clone that would grow up through life. So the process, right, it's not like a copy machine. It's still having to go and grow. It would just be identical DNA. Now, do we have clones today? Well, sort of, right? You have identical twins, which are in essence clones, okay? But that was born. They were born, of course, what we call monozygotic twins, where the fertilized eggs split. Okay, so I want to talk about 3 types of human cloning, and then we'll go into it in a little more detail. 1, therapeutic cloning. This is the idea of taking some adult cells, right? Some of the cells that are in an adult, cloning them or the proteins for use in medicine. This is a very active area of research. And if you remember, monoclonal antibodies were really big, or they may be still big, when in the COVID pandemic. Basically just cloning antibodies and then injecting them, right? So, for example, I have a little diagram of how that might work, right? You can have a particular immortalized cell, take the antibodies, let the different types of cells, actually plasma B cells are what make antibodies. It sort of makes a little hybrid here and then produces all these monoclonal antibodies. These antibodies then would go and fight the infection. So if you have a particular infection, well, we could inject antibodies that we know go and fight that particular infection. So there's really a lot of things we can start doing by cloning some of these antibodies. Okay, so the second type is what you think of when you think of cloning, right? Reproductive cloning. This is making a whole new human that is a cloned representative of another human, right? So human cloning, closer than you think, right? Time article from several years ago. This is a highly ethical issue, right? That has a lot of debate. Another one which I find really interesting is what we call replacement cloning. So this is when we would clone organs, right, and use those. Now, the benefit of cloning an organ instead of using a transplant, right? If you think about, like, if you need a kidney or if you need a liver, you get on this transplant list and, you know, someone that has passed may donate a kidney or a liver to you. But the problem is something called rejection. Our body often rejects different organs that have been put into us because, right, they're not our own organs. So if you have a cloned organ of your own liver, so if when I was for example, when I was young, I took some of my cells and I had like this big warehouse that had an extra heart, an extra liver, an extra brain, maybe. Then if one of those got damaged over time, some skin, then I could just essentially have a little warehouse. I'd go, you know, for my checkup and I'd be like, my liver's not so great. You know, I got a little heart disease. I'm ready, you know, to get transplanted. We could do a transplant and essentially, you know, live forever. The problem, of course, gets to when we get to the brain, and I'll talk about that in a moment. Okay, so I want to go into a little more specifics about how this occurs sort of molecularly and cellularly, and then we'll end with sort of that lingering ethical question. So the way we would do this is called a somatic cell nuclear transfer. And when you think of somatic, think of all the cells in the body except the gametes, right? The egg and the sperm. So basically taking any cell, right? We use mammary cells with the sheep, but basically, you know, you could use almost any cell in the body, transfer the nucleus out of it. So here's the process, right? So what you basically do, if this is the cell that has the genes we want, we would take the nucleus out and then have an egg cell, remove the nucleus from it, place the nucleus from the cell with the desired genes into it, put it through some processes, let it start cloning in a tissue sample. Then we've got a choice here. What are we going to do with this? Well, you could put that in a surrogate mother, and then the mother would carry that clone. Or if you wanted to make what we call that therapeutic cloning, remember, making some organs, we could just sort of let that sit in a tissue culture. And then hopefully, if we have the right specific transcription factors and all of those specific things turned on, then this tissue could actually turn into a particular organ that we needed. Okay? All right, so really interesting stuff going on here with cloning, but I want to end with the idea of the ethics of this whole thing, okay? I'm going to break it into two different sort of camps. One, whole human cloning. So that's taking a cell from one human, going through that somatic nuclear transfer like we just talked about, having a little baby human that then grows into an exact clone of another. So, what are the ethics of this? Well, you might say, well, what about identity? Is this, what's the identity of this person? Does it have to be the same as that person, right? Issues of identity. Is this safe? Right now, probably not. We know a lot of the clones and other animals that we make don't live very long, but that process, of course, will get better, as is everything, right? Everything gets better with time and technology generally. So we'll get to a point in humanity where we're able to do this and do it pretty well. So you have the ethical issue of just because we can, should we? Or a term that is often used in philosophy is ought we? Ought we do this thing? Okay? So I think a lot of people are kind of on the fence on this. Like, oh, I don't know, human cloning, kind of a weird idea. Another one that might pique your interest, though, is organ cloning. What about the ethics of that? Is this a route to immortality? If I, like I mentioned, just had a warehouse, if everybody had a warehouse and we went in as a child and gave a little sample so that we can make all cloned organs, could we essentially live forever? Is this a way for humanity to survive forever? Now you may say, okay, well, what about my skin? It's going to age. Well, we just cloned some skin for you too. Do a skin graft. Oh, what about my brain? That's the tricky one, right? Because if I have a brain transplant of a brain that's been sitting in a vat somewhere, that's not my brain. All the things I've learned and the experiences, that's different. Well, some scientists are proposing the idea of partial brain transplants. I like to think of the analogy as repairing a ship while at sea, right? You can't repair the whole thing at once, but you got a little bit of a board here that needs replacing. You can get a little bit of the mast of the sail up here that needs replacing. You can replace little parts of the brain over time while keeping that consciousness so that you never lose the consciousness. And with a few operations, replacing little parts of the brain over many years, perhaps I could keep my consciousness and my memory, and I'd be the same person, just with new cells in my brain, right? Crazy, crazy to think about. So you think about that for a little bit. What would you do? Are you for this? Are you for this? And then once you sort of decide where you are on this moral landscape, then I'd push you a little farther. What if it was one of your parents that was in an accident and the doctor came and said, oh, you know what? We got a cloned organ. Without this cloned organ, they're not going to make it. Do you then, when it's a little more personal, right? Do you then say, oh, my ethics have shifted a bit, right? These are tricky issues to think about, but all of this courtesy of science and biology. Pretty cool stuff.